Thank you, Aastha. And if I start pointing with the microphone and speaking into the laser pointer, someone yell at me. So it's really nice to be here. And it's also really nice to have seen this logo on quite a few presentations and posters so far. So what I'm really going to tell you is what this means, what we're doing at EBI in the imaging sphere. And Joe gave a fantastic introduction to EMBL and to EBI this morning. So I won't repeat that. Instead, I will jump straight into some of the problems. And the kind of wonderful problems are that we get images from many, many, many different sources, many technologies. Imaging is really a group of related disciplines, not just one. We get images from photons and electrons and sound waves and scanning probes. And even the photons are sometimes visible light photons and sometimes they're X-rays. And all of these things have their own challenges in dealing with the data. So what happens when we try to come together and we try to bring these things into a kind of both a unified whole for managing the data, but then potentially, and we've talked a little bit about this, a unified whole in the future for analyzing the data. And I'm not sure if you've seen this yet, but this is a figure that often gets put up when we talk about data management to kind of give us an overview of what specific goal we're trying to meet for different solutions. And what I'm really talking about is the top left part of this, preserving, sharing, and reusing. All of these things are important, but this will be mostly focused on that side. And what we do at EBI to help with that part of the process is we provide databases, and we provide databases for images, and actually also for annotations. And the word annotation is a bit overloaded, but annotation in this case, as well as, say, whole image metadata annotations. I'm talking about spatial annotations like segmentations, for example, but also point annotations are important in lots of contexts. And we do that at scales from the near atomic, so like the cryo-ET that Stephen was talking about earlier, up to tissues and organs and whole organisms, and even a little bit beyond. And these are deposition databases. So Shuichi gave a great overview of this concept of the difference between, say, the repository and a database. And another phrasing of this is that it's really useful to have a type of database that's really focused on routine, rapid publication of image data. And the typical user journey use case here is that a scientist comes with a publication, and as part of the publication process, she wants or is being asked to share that image data openly and publicly. We take the data and we take it together with some metadata, and more on that in a moment. And we put it in our databases, and then we give it back. And the reasons that we give it back are multifold, because people use data for different things. That's another reason that the things we do are challenging, because there's different downstream use cases. One of the most common and where we tend to see our data resources get their first major reuse cases are for methods development. Methods development in imaging at the moment tends to be very much about the AI cases. People want lots of data to train models, benchmark models, validate models, unify models into foundation models. We give a whole talk about this, which I won't, but that's where we see the most reuse at the moment. And the data sets that tend to be sort of hot in the sense of seeing the most views and downloads tend to be the ones that were either AI projects in the first place, so they're good for reuse or might be usable for AI. But also for training humans rather than training models. We train people in image analysis, and it's good to have kind of real world sample data for that, not the kind of Fiji blobs image, which is good for a few things, but then maybe you want to get onto what actual data look like. Or in the long run, new discovery. And again, we see that with the more mature data resources, that over time, as the data standards stabilize, you build up a big enough corpus of the data, you can then use that corpus to do really interesting new things, compare across domains, compare across images, get new knowledge. And the databases also feed more specialized resources, which can take those data either directly or do some additional processing and curation, like, for example, the cryo-ET data portal. And a lot of the examples in the cryo-ET data portal that CVII have built come from Empire, our electron microscopy imaging database. So these two main pillars almost of the work we do are focused on the reproducibility of provenance use case. So when you go to a publication, can you find the data? Can you ensure that there's something supporting the results in the publication? and then taking that and enabling new use and access to reference data on top of that. Kind of when we built the archive, we weren't quite sure how much it was going to get used, and it's turned out the answer is a lot. So these are the number of individual submissions or the number of data sets we've made available over the years. 2024 isn't finished, so the bar will be higher by the time we get to the end of the year. And this growth is, if not exponential, at least quite a lot more than linear. Roughly, this corresponds to the number of publications which we are supporting with data that you can go and download and play with. If you look at the actual data volumes, it's very, very clearly exponential. It's been growing extremely rapidly. The doubling time for imaging data that we see is under 12 months. It's the fastest growing data type across the life sciences data types like EBI. So the brief snapshot, and I've actually quite a few talks and have a sort of summary slide of where we are now. And this is the first time when I've updated this talk, I've had to change the slide during the lunch break because the numbers went up. So we got another deposition while we were eating and looking at posters. And what's also interesting is that this is the first month we've hit where we got more than one submission per day on average. So when I started, it was 3 years ago, it was about 2 a month. Now it's over 1 a day. October, we had 40-something submissions. So growth is really rapid. Where do the data come from? They come from all over the world. So there's kind of a bias to the UK and Europe because of where we're physically sited and the immediate networks. But beyond that, there's a wide range of people who submit to us. In fact, if you look, it's 43 different countries, not quite as many as we have countries represented here, but nearly there. So the data comes really from everywhere across the globe at the moment. And it comes in many different shapes and sizes and supporting many different things. So everything from, say, human embryonic development, biomechanics, through to looking at vascular patterns in flowers, through to examining genetic development in non-model organisms, through to actually using the data to do infection biology. How do we manage metadata for this? Because this is quite challenging when it comes across many different domains. We follow some principles, and I think Rembi, which again has come up in a few slides, is best thought of as a recipe for making metadata models. It tells you perhaps how you should categorize your metadata. It tells you what things are useful. It gives a great framing for conversation about metadata, because often people will talk about, do you capture this kind of metadata? Do you capture metadata? And turn out to be talking about completely different things. You could say instrument metadata about the microscope and sample metadata about the thing from the biological organism. They tend to be very different and have different requirements. So it's great framing, and it provides principles for developing a metadata model, and we have one based on this. It's aimed at being broad rather than deep because of the type of resource we are. And it's also designed so that you can link to other resources, particularly for identifiers, and particularly actually touching on the question Jan was asking about how do we start to link to other forms of biomolecular data. And identifiers, which you've heard about, are one of the things that will need to do that, I think. What this looks like in practice, here's an individual submission. And I picked this one out partly because it's a nice submission, and also because there was good quality curation work done here by Isabel in the top right. And Isabel is a data steward at BioImage. And she helped the group that produced this data submit them. And that's also why Isabel is actually included in the author list for the data set, because one of the things we're really keen on doing is making sure that data curators get credit, data stewards get credit, because we'll never make the whole ecosystem work unless we can assign credit to the people who are doing lots of the work that isn't just the primary data generation or the coming up with the hypothesis and the funding. And we have information here about the background, about what we call the study. We have information about, say, the funding and the acknowledgements, and you can spot a raw ID in the wild here, I guess, if you've been paying attention so far. There's also information then about the biological sample, about how it was prepared for imaging, and about the images themselves. And these, in a sense, get progressively more detailed as you move towards individual files. What are we thinking about next? Well, one of the things that we have to think very carefully about over the next 3 years is now we're basically at the 1 submission a day. How are we going to get to 10 per day? Because that's coming. If we look at the growth projections, it's coming, we see it, and a lot of the reason it's coming is because the really big funders, so for example, Horizon Europe and the NIH, 2 biggest biomedical funders in the world, have said, okay, you must make your data available. It's no longer should, it's now must, but haven't, at least at the broad-based level, necessarily put so much effort or maybe even thought into exactly how this will work in practice. So that's one of the reasons why we see the growth and why we're trying to catch up on this. Two things I will just highlight before wrapping up that are key to doing this for us. One is the importance of the community, which is why it's so nice to be here. So we work directly with the people that use the archives, but we also work with and through the organizations like Global Bioimaging, like Eurobiomaging, and like, say, Bioimaging UK and other national infrastructures that can really help us hear and understand what users want, but also broaden our ability to speak out to the community. This is incredibly important. And the other thing that's incredibly important is working with other players in the image data ecosystem, particularly other repositories, as Shuichi said, that are working and solving similar challenges so that we can do the best we can together for the community. Another key pillar, and you've heard quite a bit about this so far, is on the technical side. So OMI NGFF has really been a game changer for biological imaging. So we heard earlier about how, say, for some of the more medically focused side, everything can be in DICOM. That is not the case and can't really be the case for biological imaging. The OMI NGFF holds some promise for standardization in bioimaging. And one of the ways we are supporting, we convert an increasing quantity of the data we come in that comes in incompatible formats into OMI ZAR. And we do that so it can standardize going out. And actually standardize going out is important in some sense before standardized coming in, because that's how we demonstrate reuse. But it's also how we can do things like enable visualization. So this is kind of actually working, if anyone wants to see this, you can play with this, I can give you the link. This is a working preview of what almost the BioImage Archive 2.0, the next version of our website, will look like. And this, the great thing about this is you actually get in-browser visualization of all of the things we've managed to convert. And because of the wonderful things about chunk pyramidal formats, you can view these enormous slide scanning images, and you can go and look at the things that are interesting to you. But you can also stream the data. So you can grab the link for that. You can open it in Napari, look around in Napari. You can open it in Fiji. You can access it via the API. If you want to do machine learning, you never want to see a pixel, you can get the data programmatically, run model training on it. Again, standard format, lots of viewers. You can pick an image, and rather than look at it in a 2D plane viewer, you can go and look at it in 3D, which is great. Doesn't work for everything, but a number of the images we have work well in 3D rendering with no meshes. So these things, getting the right community or getting the community together, being part of that community, and getting the right technical foundations are the things that will help us go forward for the next few years and really scale up. And now I'll stop. I'll thank my team and our funders, without whom none of this would be possible, and happily take whatever questions we have time for.
Thank you so much.